Hello, my name is Baraa Orabi. I'm a PhD student at Dr. Faraz Haj's lab at the Department of Computer Science at the University of British Columbia, and I'm presenting to you our work under the title Annotation-free Isoform Discovery using Long-Read Sequencing. So before I start, I would like to talk a bit conceptually about the problem of isoform identification versus isoform discovery. So when we talk about identification, what we typically mean is that you have a set of predefined isoforms from some annotation database, and you have your sequenced reads, and you're trying to label those reads or identify the isoform of each of these reads. However, if you have some novel isoform, you're most definitely going to mislabel some of your reads. On the other hand, when it comes to discovery, what we're trying to do is that we're starting from the reads and trying to minimize or eliminate the annotation reference bias and trying to identify the structure of the isoforms that we have in the reads without reliance on this annotation database. So isoform identification and discovery, when it comes to using long-read sequencing, is much more powerful technique. This is because of the potential for full-length transcript sequencing and the ability to work with even single samples. However, this is not a trivial problem. This is because of the high sequencing error rate and sequencing structural artifacts that are common in long-read sequencing. So Freddie, our method here, is an annotation-free isoform discovery tool using long-read sequencing. Freddie has three main stages: split, segment, and cluster. And I'm going to get into the details of these different stages. But first, the input to the whole tool is genomic splice alignments. And just to point out, at no stage in Freddie do we require isoform annotations as input. And I think this is a true strength of our computational tool. So first, let's talk about split. So the goal here is to partition the genome into sets of intervals such that no two sets are linked by any read alignments. So for example, over here, we see that this interval over here is linked to this interval by two reads, which is in turn linked to this last interval by two other more reads. So once we computed all of these intervals or sets of intervals, each of these sets can further be processed independently from the rest, which allows us for highly parallelized computation in Freddie. So next step is segmentation. And here the goal is to segment each interval into canonical segments. So what do we mean by canonical segments exactly? Well, assume that you have a set of underlying isoforms, as you see here. And the canonical segments, they need to be able to represent any isoform by just mere presence or absence of each of these segments. So in a way, it is the union of all of the exon boundaries of your isoform set. So how do we do this from the reads without reliance on the annotation? Well, first, for each position in each of our intervals, we count the number of reads that either start alignment or end alignment in each of their respective positions. This is not the same as coverage, because here we're only looking at the start or end of each alignment interval for a given read. Then we have the signal. We smooth it out using just a Gaussian filter. Next, we identify the peaks, and we identify them as candidate segmentation boundaries. So we get so many of these, and we want to be able to minimize their numbers. So the way that we do this is that we want to select a subset of these peaks and call it S, that maximizes a splicing signal's core function, which is this core function over here. And in short, what this does is that it takes each couple of segments, and then it tries to minimize the number of reads that are ambiguously or cannot be represented well for a given segment. So you want to minimize that number, and you want to maximize the number of reads that supports the boundary between two consecutive segments. We're able to identify the set S that optimizes this function using dynamic programming formulation. So once we have this, we're now able to represent each of our reads as a binary fixed-sized vector. And this is very cool because now you're able to compare two different reads using meaningful subunits, which are the canonical segments, in very efficient and useful manner. So after this, we do the clustering. So in clustering, just an overview, we want to cluster our reads into isoforms. However, we don't know how many clusters we have because we're not using any annotation. And we don't want to overfit because we know that the underlying technology has noise. So what we came up with is a unified model that addresses these two challenges together. And we call this minimum error clustering into isoforms or MERCI problem. So the way that this works is that we have an isoform bin and a recycling bin. And the idea is to partition the reads into these two bins such that we minimize the number of reads in the recycling bin and minimize the number of errors of the reads in the isoform bin relative to a consensus that they form within each other. So in the first iteration, we optimize this using integer linear programming. And now we set aside the isoform bin reads, and then we recycle the reads in the recycling bin into a next iteration. We keep doing this until reads are exhausted or the isoform read support falls below a certain set threshold. So as preliminary results, wanted to show you this sequencing experiment that we've done. So this is on 22RV1 cell line. This is a prostate cancer cell line. And we find two potentially novel isoform not identified before in the major annotation databases. This finding is not yet confirmed by orthogonal technologies, so we would like to do this. And with that, I would like to thank my supervisors and the whole team at the Vancouver Prostate Center and in our research group. So thank you so much, and any questions?